Friends, welcome to this YouTube channel. Today we are going to discuss a topic in developmental biology, that is gametogenesis. This topic is dealt in two parts. The first part is dealing with the spermatogenesis and the second part is dealing with oogenesis. Okay, let us get started. Friends, you may be knowing very well that the developmental biology is the study of the development of an organism from the egg stage to the adult stage. During this course of development, there are several steps involved in it. They are the gametogenesis. Gametogenesis is the process of production of gametes. Copulation. It's a process in which the male and female unite with one another or copulate with one another. Fertilization. It is a process in which the two gametes, a male gamete, sperm, and the female gamete, egg, unite with one another. Cleavage. Cleavages are the phenomenon in which the zygote undergoes divisions, specific divisions, and later morulation, blastocyst formation, implantation, a close intimate contact with mother and embryo, and then formation of bilaminar embryonic disc, gastrulation, trilaminar embryonic disc, notochord formation, nerve cord formation, mesoderm differentiation, extraembryonic membrane formation, placenta, and properties of gestation, parturition. All these are the events that we need to study in order to understand the developmental biology of a particular organism. Okay, today's topic is gametogenesis. Gametogenesis is of two types. One is called the spermatogenesis, in which the sperms are formed, and the other one is called oogenesis, in which eggs or ova are formed. Okay, so gametogenesis, or the reproduction, let us say sexual reproduction, is a continuous process, or this process is needed for the perpetuation of a species, for the continuity of a species with a lot of genetic recombinations. You know that sexual reproduction is essential. Sexual reproduction is nothing but union of the gametes of two different parents, the male parent and female parent. The phenomenon in which the gametes are formed is called today's topic gametogenesis. Okay, so our body is built in with the two different types of tissues. One is called the somatoplasm, which is meant for our own survival. You take, for example, our eyes, nose, ears. These are all meant, our digestive system, respiratory system, excretory system. These are meant for our own survival. But there is another tissue called germinal tissue. The germinal tissues develop gametes, produce gametes, and these gametes are having half the number of our chromosomes. In order to maintain the chromosomal number constant in the entire race, they carry our genome and they mingle with, or they meet with, or they fuse with the other gamete, that is the gamete of opposite sex, and forms a zygote. So that what happens, the chromosomal number will come to the normalcy and the process will go on. So that's why the somatic tissue is meant for our own survival, and the gonadial tissue is meant for our further generations. And you may be knowing that there is sexual dimorphism. Male and females are having separate external features. Internally they are having separate gonads. Males are having testes and the females are having ovaries. So externally some morphological characters differ in males and females. By that way we can easily identify. Okay, germ cells are located in gonads, and germ cells are almost all cuboidal cells, and the germ cells are very important because any change that happens in the germ cells will result a change in the next generation, and they are going to produce the gametes. What is gametogenesis? Gametogenesis is nothing but gamete genesis, production of gametes. There are two types of gametes, as I mentioned. Males produce gametes called sperms and females produce the gametes called ova. Hence, gametogenesis can be studied under two headings. Production of sperms, that is called spermatogenesis. Production of ova, that is called oogenesis. And you know very well that the entire process of this kind of reproduction, events in sexual reproduction, are regulated by some certain chemical messengers. They are called hormones. The hormones that are secreted from our hypothalamus regulate the pituitary. In turn, pituitary regulates gonads. So that the entire process of sexual reproduction is under the control of our hypothalamus with the help of certain chemical messengers. And what is very important what is the very important thing that occurs during gametogenesis is the recombination. Say we are having chromosomes in pairs. These pairs of chromosomes, you take for example a specific number, chromosome number 5 is having a pair. In this pair, one chromosome has come from father and one chromosome has come from mother. During the gametogenesis, a special division that occurs is called meiosis, reduction division, and the prophase of reduction division is very, very special, and during this prophase recombinations occur. So the genes what are there in father chromosome will go to the mother chromosome and vice versa occurs. So this kind of recombinations result in meiosis, and that meiosis occurs during gametogenesis. As a result of that, very wonderful blending of father-mother characters will appear in the next generation. The characters of father and mother nicely blend, okay, perfectly blend, and during recombinations, and we will see their expression in the next generation. So let us start our first part of this video, and after that the second part will continue. That is called spermatogenesis. Okay, spermatogenesis meaning formation of spermatozoa from its mother cells called spermatocytes. Spermatocytes are called sperm mother cells. You will see here cuboidal cells, and all these cuboidal cells are coming from the germinal epithelium. And where is this germinal epithelium? In special tubes called seminiferous tubules. And where are these seminiferous tubules? They are situated in testicular lobes, and this is called testis. Testis is a male gonad. Testis is the male gonad, and this is covered with the certain layers called tunicolbugenia, and the tunicolbugenia goes inside the testis, forming several testicular lobules, and these testicular lobules are provided with the small coiled and supercoiled tubules. These tubules are called seminiferous tubules. The seminiferous tubule is having, you can see here, it is having a lumen and it is having the germinal epithelium. The wall of seminiferous tubule is having germinal epithelium. Okay, along with this germinal epithelium, there are some special cells called Sertoli cells present. And outside the seminiferous tubules, in between the seminiferous tubules, and there are certain other cells called Leydig cells, interstitial cells we call. So these three cells are taking a very vital role in spermatogenesis. The germinal cells, the Sertoli cells, and the interstitial cells, otherwise called Leydig cells. Where are the germinal cells present? These are present in the seminiferous tubules. Where are the seminiferous tubules? They are located in the lobules, testicular lobules of testis. Okay, so let us observe what is that called a seminiferous tubule in this diagram. Okay, this is a certain portion of seminiferous tubule inside the seminiferous tubule. Okay, you see here this is the epithelium, and you will find here several cuboidal cells. These cuboidal cells are the stem cells. They are called spermatocyte or spermatogonia. They are called spermatogonia. Okay, they are called primordial germ cells. The primordial germ cells are formed during the embryogenesis itself. During the embryonic development, the gonads will have certain cells, and those cells are called primordial germ cells. By the side of these primordial germ cells, you can also locate the presence of other cells called Sertoli cells. Okay, so these spermatogonia, they undergo certain steps to form the sperms. Okay, these are the sperms, matured sperms. They are formed from some cells. Okay, resulted in the divisions of these primary spermatocytes. They are called spermatids. Certain division occurs here, very special division, that is called meiosis. Okay, meiosis 1, meiosis 2, that occurs. As a result of that, from one primary spermatocyte, 4 matured sperms are formed. But friends, you just understand, the spermatogenesis can be studied under 2 headings for our convenience. One is called formation of spermatids. From the primary spermatocyte, in the next slide I am going to explain. Okay, from the primary spermatocyte, after reduction division, 4 cells are formed. They are called spermatids. What is the difference between a spermatid and a primary spermatocyte? The difference is there in the chromosomal number. Primary spermatocyte is diploid, whereas the spermatid is haploid. Primary spermatocyte diploid, spermatid is haploid. Okay, so this is first stage. What is this first stage? Formation of spermatids. The second stage is very important. In the second stage, what happens? A cell now differentiates into a spermatozoa. It will attain certain parts. Say a head part, acrosome, a neck part, a middle piece, and a very long flagellum tail. So all these parts are formed by another very important process. So the formation of spermatozoa from spermatid is called spermiogenesis. Sperms are produced. Sperms are produced from those unspecified cells, undifferentiated cells. So friends, spermatogenesis has 2 steps. One is called formation of spermatids. The second one, conversion of spermatids into spermatozoa. Of course, the second process is called spermiogenesis. Okay, let us study these 2 processes. But in the first process, what is the first process? Formation of spermatids. That includes 3 stages further. One is called a multiplication phase, a growth phase, and the third one is called maturation phase. Okay, let us see. So let me explain this. This is the testis, and on the testis there are the rete testis, small vasa efferentia. This is epididymis. Okay, epididymis leads into a tube called vasa deferens. Okay, inside the testis there are several coiled tubules called seminiferous tubules. Inside the seminiferous tubules, spermatogenesis occurs. Okay, spermatogenesis, let's see. Inside the seminiferous tubules there are several stem cells. These stem cells are called spermatogonia, whose chromosomal status is diploid, that is 46. The spermatogonia undergo multiplication phase, and during multiplication phase it undergoes mitosis. With the help of mitosis, one spermatogonia forms many cells, and in this process, during mitosis by spermatogonia, 2 different types of cells are actually formed. They are classified as type A spermatogonia, type B spermatogonia. Type A spermatogonia will be renewing as spermatogonia and settle there itself for further spermatogenesis. And type B cells are presently used to produce the sperms. So at this particular time, sperms are produced. not from all the spermatogonia, only from some spermatogonia which are called sperm precursor cells. They are designated as type B cells. Type A cells remain there itself for further spermatogenesis. Okay, during multiplication phase, as the spermatogonia perform mitosis to form number of cells. From these cells that are formed by mitosis, some cells will be retained there itself to act as further spermatogonia. They are called type A cells. And few cells are descended down to produce the sperms for now or for this particular time. These are called type B cells. So let us call the type B cells as primary spermatocytes. Let me tell you, spermatogonia divide by mitosis to form primary spermatocytes and spermatogonia. Okay, primary spermatocyte, because it is produced by mitosis, it is also diploid. It is also diploid, having 46 chromosomes in human being. So this primary spermatocyte undergoes growth phase. Okay, it will consume some other cells, or they'll be nourished by some certain Sertoli cells and other cells, and they gradually grow in size. This increasing in size is called growth phase. Which undergoes growth phase? Primary spermatocyte undergoes growth phase. What is the chromosomal status of primary spermatocyte? Diploid. And how is the primary spermatocyte formed? By mitosis from spermatogonia. Okay, the primary spermatocyte now undergoes the next phase called maturation phase, in which maturation divisions occur. The maturation divisions are nothing but meiosis. Primary spermatocyte undergoes first meiosis. You know very well, first meiosis is the reduction division. It is the reduction division. During first meiosis, the chromosomal number is cut to half, so that the primary spermatocytes undergo first meiosis to form secondary spermatocytes. The secondary spermatocytes are haploid in status. They are having only 23 chromosomes. So reduction division occurred in secondary spermatocyte. The secondary spermatocytes now undergoes meiosis 2, which is nothing but a mitosis. As a result of that, each secondary spermatocyte forms 2 spermatids. In total, as there are 2 secondary spermatocytes from one primary spermatocyte, okay, 4 spermatids are formed from one spermatocyte. 4 spermatids are formed. What is the chromosomal status of spermatid? It is also haploid, because this is mitosis. Meiosis 2 is nothing but mitosis. So this, up to the formation of spermatids from primary spermatocyte, it is called formation of spermatids. Okay, now let us see the second phase. What is the second phase? How the spermatocytes are converted into sperms. That is called spermiogenesis. Spermiogenesis is a process through which undifferentiated spermatids will be converted into very specially shaped spermatozoa. And after the spermatozoa are formed, they are released into the tubules, and from the tubules they go to the epididymis by a process called spermiation. Let us see the spermiogenesis. Okay, the spermatogenesis 2. This is explained growth phase, and this is called maturation phase. Okay, I have already explained this. Let us see the next phase, that is called spermiogenesis. What is spermiogenesis? Formation of spermatozoa from the spermatids. How the spermatids transform into spermatozoa. This is called spermiogenesis. Okay, so the transformation of spermatids into spermatozoa is called spermiogenesis or spermatogenesis. Okay, each spermatid transforms into one sperm. From each spermatid, only one sperm is formed. As there are 4 spermatids from one primary spermatocyte, 4 sperms are formed. Okay, so right. The Golgi, you see here Golgi. The Golgi in the spermatid is going to form acrosome. Golgi apparatus forms acrosome because acrosome is the structure of the sperm which is provided with the enzymes to penetrate the egg membrane. So what forms the acrosome? It is nothing but the Golgi of spermatid. Okay, the centriole, what is present here? Of course, the centrioles are of 3 types here: the proximal centriole, the distal centriole, and also the end centriole we call the circular annulus. So here the centriole is going to form a long flagellum which later modifies into what is that called a tail. Okay, then several mitochondria are there, These mitochondria are going to produce energy. Okay, they are energy and they are stored in the middle piece. They are stored in the coiling apparatus of the middle piece, and so that is the powerhouse of the sperm. And the cytoplasm is you see in spermatid lot of cytoplasm is there, but in sperm very less amount of cytoplasm is there, indicating that some of the cytoplasm is thrown out, some of the cytoplasm is shed off. Okay, then gradually head is developed, and the head of the sperm is found embedded in the Sertoli cell, because Sertoli cells are called the nurse cells. They are meant for nursing the sperms. They are meant for providing nourishment. Where are these? These are located by the side of the what is that called germinal cells. Okay, and they are formed. They are activated by certain enzymes like certain hormones like FSH. We are going to see the hormonal control also at the end. Okay, now some cytoplasm is shed, head becomes embedded in the Sertoli cells, and they go to the lumen, and where from they are released into the tubules. This process of liberation of sperms from the germinal epithelium into the tubules is called spermiation. Flow of sperms, that is called spermiation. Okay, now one thing we need to understand, that is the spermatogenesis occurs in 2 stages. In the first stage, formation of spermatids. In the second stage, transformation of spermatids into sperms. And the formation of spermatid occurs in 3 phases: multiplication phase, growth phase, and maturation phase. During the maturation phase only meiosis occurs. The meiosis in the case of spermatogenesis occurs continuously. Meiosis 1 followed by meiosis 2. Okay, meiosis 1 and 2 occurs one after the other. First meiosis 1, later meiosis 2. And this is the difference you will notice if you study, if you observe the oogenesis. In oogenesis, meiosis 1 takes several years to complete. Here, the entire process of meiosis 1 and meiosis 2 and what is that called spermiogenesis will be completed within 5 weeks of time. Within a month, the sperm will get matured and is formed. But whereas egg is developed by taking some years of time. Okay. So during the embryonic development, gonads will receive some cells. These cells are called primordial germ cells. The primordial germ cells remain within the gonads. They remain within the gonads and they don't divide by the time. Okay. So whenever a boy gets the age of puberty, reaches the age of puberty, then the onset of sexual maturity occurs. By that time, these cells, primordial germ cells, remain calm. Later, after the puberty, they'll be activated by the hormones. Okay, right, here there is a small spelling mistake. Okay, right. Next, let us see the next one. How the hormonal control of spermatogenesis occurs. We know very well that hormones are playing a very, very important role in controlling both oogenesis and spermatogenesis. And what is the master gland here? The master is hypothalamus. Hypothalamus, after the onset of puberty, when the age of puberty comes, the hypothalamus releases GnRH, gonadotropic releasing hormones. Gonadotropin releasing hormones are released through the neurohypophyseal system into the pituitary gland. The pituitary, especially the anterior pituitary, adenohypophysis, anterior pituitary gets stimulated by these GnRH, and immediately anterior pituitary secretes luteinizing hormone and follicle stimulating hormone. Two hormones are liberated. One is called the luteinizing hormone and the other one is called FSH, follicle stimulating hormone. Let us see what is their role. Luteinizing hormone influences the interstitial cells of testis. Hence, luteinizing hormone is also called interstitial cell stimulating hormone. This is called ICSH, interstitial cell stimulating hormone. What it will do, it will stimulate the androgen secreting cells, endocrine cells called Leydig cells, to secrete testosterone. Okay, when testosterone is secreted, then the boy develops secondary sexual characters, facial hair, axillary hair, pubic hair, all those under lowering of voice, okay, decrease in the pitch of voice, all that are male secondary sexual characters, they are developed by the influence of testosterone. Testosterone is secreted by Leydig cells of testis by the influence of LH secreted by pituitary gland, and that pituitary secretes LH by the influence of GnRH comes from hypothalamus. So complete control of the spermatogenesis or the sexual reproduction is there with the hypothalamus. Right. What is the role of FSH? This is very, very important. Anterior pituitary secretes follicle stimulating hormone. But you know very well there are no follicles in the case of male. But instead of stimulating follicles here, you need to understand the adjacent cells. The adjacent cells are nothing but Sertoli cells. So follicle stimulating hormone stimulates the Sertoli cells. These Sertoli cells, they secrete two hormones. One is called androgen binding binding hormone, and the other one is called inhibin. Okay, androgen binding protein, that is actually called androgen binding protein. What is secreted by the Sertoli cells makes the testosterone liberated by the Leydig cells get concentrated at a particular place. Okay, testosterone is produced by Leydig cells, but this testosterone is to be concentrated at a particular place. So androgen binding protein, when it is there, then testosterone will show its effect. Otherwise, under normal conditions also, by what is that called adrenal gland, some sex corticoids will be liberated, but they are not aggregated at a particular place. But here what happens, there is androgen binding protein secreted by a Sertoli cell, makes the entire androgen secreted by the Leydig cells get concentrated at a particular place in order to stimulate the spermatogenesis process. And the second protein what is released from Sertoli cells is inhibin. Inhibin is a very, very important protein. Inhibin will inhibit FSH activity. It will inhibit the FSH activity, hence it is called inhibin. When does it inhibit? It will inhibit the FSH activity whenever there is more sperm count in the body. when there is more sperm count in the body and there is no need of further production of spermatogenesis, then immediately the Sertoli cells recognize the sperm count, okay, sperm count, and immediately it will stop FSH function. When FSH function is stopped or inhibited, no ABP, antigen binding protein, is produced, so that testosterone will not get accumulated. So that spermatogenesis will be ceased for some time. But when sperm count becomes low, the sperms are already ejaculated, then the sperm count becomes low, then Sertoli cells will not release inhibin. When there is no inhibin, FSH will act upon the Sertoli cells. How beautiful, what is that called, control or feedback mechanism is there. And this kind of feedback mechanism, we say the negative feedback mechanism, because the Sertoli cells are negatively affecting the anterior pituitary, hence it is called negative feedback. Once inhibin is liberated, automatically the hormone level goes down. Negatively it is affecting. That's why it is called negative feedback. Okay. So what we have seen, the effect of FSH on Sertoli cells, ABP, and also inhibin. Inhibin will inhibit anterior pituitary to secrete what is that called FSH, or FSH activity will be inhibited. And then you see, when Leydig cells are releasing testosterone and the testosterone got accumulated at a particular place, this is called rising level of testosterone. When rising levels of testosterone occur in the blood, immediately the testosterone themselves negatively feedback hypothalamus as well as anterior pituitary, so that LH secretion is also regulated. So LH and FSH are playing a very, very important role in spermatogenesis. LH is stimulating, what is that, the Leydig cells, otherwise called interstitial cells of testis, to produce the male androgen testosterone. FSH is stimulating the adjacent cells of the germinal cells. They are called Sertoli cells or nurse cells. And the nurse cells play three very important functions. One is they nourish the sperms. The second one, they secrete androgen binding protein ABP. They also secrete inhibin. ABP will accumulate the testosterone. Inhibin released only when there is more sperm production. To cut down the sperm production, inhibin is liberated, and of course it will inhibit FSH action. And the third one, what is very, very important, is that nourishment. Again, I have explained. So this is the beautiful control of hormone over the sperm production. This is called hormonal regulation of spermatogenesis. Friends, after having studied the spermatogenesis, it is very important to see the structure of a sperm or spermatozoa. Okay, a sperm is very much microscopic. It is less than, what is that called, here it is given, it is about 0.1 millimeter. So we cannot see it with our naked eye. We need a magnifying lens or a microscope. But egg liberated by female can be seen by our naked eye. Okay, you see the photograph or you see the diagram of a sperm, human sperm. What does it have? The sperm is having head, a small neck, middle piece, okay, and tail. Tail is further divided into main piece, end piece. End piece where there is no sheath is there over axoneme, and then here axial filament this is, and there is a protoplasmic sheath that is called the main piece, and this is called middle piece, this is called head. Head is consisting of very less amount of cytoplasm, almost all occupied by the entire nucleus. But on the top of the head you will see a cap-like structure. It is known as acrosome, a very important structure. Acrosome is having enzymes, especially hyaluronidase, an enzyme. Hyaluronidase will penetrate the egg membrane, and several proteolytic enzymes are there. Acrosin, another enzyme needed for penetration of corona, and hyaluronidase needed for the penetration of zona, and these are called spermilysins because they are the lysine enzymes present in sperms. So they are used to contact and penetrate the ovum. If a sperm is having defect in the acrosome, it may not be able to penetrate the egg. Okay, that is called a defective sperm, and it will never become fertile. Okay, this is called head, and just beneath the head there is a small depression that is called neck. Okay, that's called neck. It is very short between egg and middle piece. It is having a proximal centriole and a distal centriole. Proximal centriole is playing a very, very important role in the cleavage of egg. That is the key. Okay, whenever the sperm goes inside the egg, then egg starts cleavages. So where is the key of cleavage of eggs? It is proximal centriole. Okay, the distal centriole is going to form the axial filament, that is the base for the tail swimming. Okay, proximal centriole needed for the cleavage of egg and cleavage of zygote. Distal centriole is needed for the formation of tail. Okay, this is middle piece. This is called a nebenkeron. It is having a coiled structures of mitochondria, and mitochondria are needed for generating the energy for the movement of sperm. And then at the end of the middle piece there is another centriole, and this is called ring centriole. The function of ring centriole is not known. Okay, and the tail is meant for swimming, and the tail has two regions. One is called main piece, occupied by a sheath, and the other one is called end piece, where it is naked. Axial filament is naked. Okay, now the sperms are measuring about 5 micrometers, 0.05 millimeters, so they cannot be seen under normal eye. We need to observe it under a microscope. Okay, but egg is 30 times bigger than the sperm. Egg is 30 times bigger than sperm, and then the sperms move very slowly, actually some millimeters per second in the female genital tract, and they can survive for 24 hours to 48 hours depending upon the surrounding conditions of female genital tract. And after reaching into the female genital tract, within 5 to 6 hours of reaching, so they get a capacity to move. Okay, that process is called capaciation, and capaciation is caused by certain enzymes released by the female genital tract. It simply dissolves the cholesterol cover of the sperms. Okay friends, this is about the spermatozoa. Okay, what is the number of the sperms that are given out by father? In a single ejaculation of semen, 200 to 300 millions of sperms are ejaculated. But when a person is said to be a fertile father, out of the 200 to 300 millions of sperms, if 60% of the sperms are having normal shape, head, neck, middle piece, tail, like that, a normal shape is there, at least for 60% of the sperms that are ejaculated, and of 60%, at least 40% of the sperms must have a good motility, a vigorous motility. Then only such a semen is said to be a fertile one. Otherwise, fertilization problems occur. These sperms cannot fertilize the ovum. Okay, and then these sperms are liberated or released along with certain other juices, and those juices come out from the seminal vesicles, the vasa deferens, the epididymis, and certain other glands like prostate gland and the bulbourethral glands. Certain other glands are there. Okay, all together mixed up in a solution called semen, in which the sperms are there, and alkaline acidic conditions are also there to make the sperm active. Okay friends, hope you have understood today's lesson. What we discussed is about the spermatogenesis. Spermatogenesis is the production of sperms, okay, from spermatogonia. Spermatogonia produce primary spermatocytes by mitosis. Primary spermatocyte undergoes first meiosis to form secondary spermatocyte. Secondary spermatocyte undergoes second meiosis to form spermatid. And the spermatid transforms into sperm by a process called spermiation, and the sperms are liberated into the tubules by spermiation. Spermiogenesis. Spermatids are converted into sperms by spermiogenesis, and sperms are released by a process called spermiation. Hope you have understood spermatogenesis. In our next video lesson, we are going to discuss the next gametogenesis, that is oogenesis, which occurs in female. Okay, it will be continued in the next video lesson. Thank you. Thank you for watching.